Good afternoon, everybody. And I think Professor Sopory has joined.
Good afternoon, sir.
Good afternoon, sir. Good afternoon, Professor Sopory.
Good afternoon, sir. So shall we start or wait for 5 minutes, sir?
I think we can wait for 2 minutes.
Okay, sir.
Mm.
Good afternoon, Dr. Maiti.
Good afternoon, sir. Good afternoon.
Great to
Namaskar.
see you.
How are you, sir? How are you?
Sir, namaste, sir.
Great to see you.
Is it 3 o'clock just, or we are
No, sir. We have to wait 2 minutes.
2 minutes? Then we will start at, let's start at 3 o'clock. And okay.
Good afternoon, Dr. Mathi and Dr. Sewar from India.
Good afternoon. Good afternoon. Very good afternoon. How are you?
You are showing that slide, no? Please,
This is the
pro. Please remove the pro. I know Dr. Sopory. I know Professor Sopory since 20, more than 20. Yes.
Good afternoon, everybody.
Good afternoon.
Prasad.
And I am, sir, I am lucky my one book chapter published.
You're on mute now, I think.
Sir, good afternoon, sir, Professor Sopory.
Yeah, good afternoon. Good afternoon.
Sir, my one book chapter with Professor P. L. Unya in North Campus, University of Delhi.
Right.
We published one book chapter with your book. And we know, I worked for 10 years in Forest Department and Environmental Biology. And
Right.
I joined, sir, 2010 year direct senior scientist in NVT New Delhi.
Right.
Thank
Great.
you very much.
Great to see you.
Shall we start, sir? Now it's
Yes,
time.
please start. Please start.
Over to Sutapa.
Very good afternoon, sir. Respected Dr. Dipankar Maiti, Director, National Rice Research Institute. Our today's speaker, Dr. Sudhir Sopory, sir, Pro Vice Chancellor, Jawaharlal Nehru University, New Delhi, and presently the chairman of our Institute Research Advisory Committee, all heads of the divisions, scientists, and personnel from National Rice Research Institute and other ICAR institutes, ladies and gentlemen. A very good afternoon and a warm welcome to the Platinum Jubilee Lecture Series. Heartiest welcome to one and all. Though our today's speaker is one among us, but I will take the opportunity to brief Sir about the Institute and the lecture series. This prestigious institute, sir, was established in the backdrop of the horrible 1943 famine known as the Great Bengal Famine. The famine caused around 2.5 million deaths due to starvation. The cause of the disaster that time was not a human disease like in today's era of crisis, but was a disease of rice plant called the blast disease caused by Helicase Pathway that led to a serious shortage of rice grain in the Bengal province, causing the Great Bengal Famine of 1943. The then government of India realized the importance of rice in the area and decided to intensify research on all aspects of rice crop. As a result, government established the Central Rice Research Institute on 23rd April 1946. Dr. K. Ramaya, who was an eminent rice breeder, was its founder director. Central Rice Research Institute has been renamed as National Rice Research Institute in recent past, that is in 2015. Since then, the institute, since 1946, its establishment has been tirelessly working on all researchable aspects of rice crop and has made significant contributions towards enriching the rice science and ensuring food security of the country. The seeds of grain revolution were first planted at the fields of this institute and spread to the rest of the country. Till now, the institute has released 148 varieties, developed numerous crop production technologies, multiple methods to safeguard rice from disease and pests, and continuously provided assistance and models to the rice farmers to make the farming more profitable. National Rice Research Institution has got a total of 5 divisions: the Crop Improvement Division, Crop Production Division, Crop Protection Division, Crop Physiology and Biochemistry Division, and Social Science Division. We also have 3 regional stations located at 3 different states of India. Additionally, 2 Krishi Vigyan Kendras are also managed by National Rice Research Institute. The first brick of the institute was laid in 1946, and we are completing 75 years of our glorious presence in the year 2021 in the country. As part of the long celebrations, National Rice Research Institute is also organized Platinum Jubilee Lecture Series. These lectures are delivered by outstanding personalities of the country and abroad. The first lecture had been delivered by Fellow of Royal Society, Professor Pitt Smith from UK. The second lecture was delivered by Swami Atma Priyamji. Pro-Chancellor of Ramakrishna Mission Vivekananda Educational and Research Institute, Kolkata. Peter Douglas had delivered the third lecture, Geographical Indications, or GM. The fourth lecture was delivered by Professor Puruganan on the long debated topic of the origin of rice. The fifth talk in the series was delivered by Dr. A.K. Singh, which was on the innovations and initiatives in the agricultural extension systems in India. And today, sir, we are eagerly wait to listen to you on the Helicase pathways in rice, role in biotic stress tolerance. Now, without taking that much time, I would like to request our director, Dr. Dipankar Maiti, to share today's event. I request Sir formal welcome and introduce today's speaker to the audience. Please, sir, over to you.
Thank you, Dr. Sutapa. I request Dr. B.C. Patra, the chairman of this lecture series organizing committee, to welcome and introduce the speaker to the audience.
Thank you, sir. Respected chairman, respected for guest of this evening. Pleased to introduce the speaker today. He is none other than our Professor Sudhir Kumar Sopory. He was former Vice Chancellor of Jawaharlal Nehru University, New Delhi. Biodata says like this: he was born in Ambala town in Haryana in 1948. Got his education from Srinagar, from ST College, Srinagar, 1962 to 66, and did his MSc from Kashmir University in 1968, and then he Delhi for his PhD program from University of Delhi, 1970. Later, he joined as a postdoc fellow in different foreign institutes like Max Planck Institute in Germany from 76 to 78, University of Texas, USA from 1981 to 82, and USDA Wellsville, USA from 87 to 88. And later, he also moved to the University of Munich, Germany from 91 to 92. If you see his service record, he joined uh, Jawaharlal Nehru University as assistant professor in 1973 to 79, and became associate professor from 79 to 85. Later, he was promoted to professor of plant physiology from 1985 to 97. Later again, he moved to International Center for Genetic Engineering and Biotechnology, ICGB, New Delhi, as head plant molecular biology from 1997 to 2011. And finally, he became Vice Chancellor Jawaharlal Nehru University, Delhi from 2011 to 15. After that, he became the interim director of ICGB from 2014 to 15 as an interim director. And since then, he has been working there as a senior emitter scientist from 2016 to till date. His basic research areas are molecular plant physiology, stress biology, and plant biotechnology. He has several awards and accolades to his credit. He has been awarded with ISCA Hiralal Chakravarti Award, ISCA Birbal Sahani Centenary Award, CSIR Shanti Swarup Bhatnagar Award, NAS Saligram Sinha Medal Award, American Society for Plant Biologists 2010, President Indian Society of Plant Physiology from 2013 to 16, and the most coveted award of Padma Shri Award. He was awarded with Padma Shri Award in 2006. And apart from that, he has been awarded with several fellowships, like he is the Fellow of Indian National Science Academy, Fellow of National Academy of Agricultural Sciences, Fellow of Indian Academy of Sciences, Fellow of Academy of Sciences for the Developing World, TUAS. So we are fortunate to have you, sir, with us today. He is also our chairman, Research Advisory Committee for NRRA from 2020 to 2022. So we had, just last year we had with him, he was chairing our session. It was excellent function last year. And sir, soon we'll be coming to this year also. We'll probably, if things move, we can see you in our headquarters at Cuttack also. We'll have it in physical mode. With this brief introduction, I will request our honorable director to invite the speaker of the day and start the proceedings. Thank you.
Thank you, Dr. Patra. Sir, may I now request you to take over the dais?
Thank you. Thank you so much. I think, let me share the screen. I did some practice. In case I some problem, I will get. Can you see my
Yes,
slide?
yes, sir. Yes,
Yes,
sir.
sir. Yes, sir.
Just put
Yes,
on the
It's visible,
slide
sir.
show mode. Yes, sir. Yes, yes.
Yeah.
Yes, sir. You can start, sir.
Oh, great. I succeeded, because in the morning I had to do some practice. I'm not used to this for the first time. I'm going through this online mode of presentation.
Yes, sir.
Okay, Dr. Methi, Dr. Patra, all the fellow scientists of NRI, the students, the staff, and it's very difficult in this mode to find out who others are there in that, but all the heads of the divisions and people from ICR, if there are any. It's actually great pleasure for me. Very happy to participate in this Platinum Jubilee celebrations of NRI through this lecture. Not to mention that NRI has, through dedication of many scientists through all these 75 years of its existence, and also through the great leadership which has been provided by different directors, the present director, the previous directors, and has made seminal contributions in rice research, which have actually made very high impact in the field of agriculture, as well as great impact in the society at large in this country. And his contributions are innumerable. And therefore, at outset, I would wish all the best to this institute, to all of you, for all your future endeavors. And the country look forward to your contribution in meeting the demand of food as well as nutritional demand in this country. Now, today in this lecture, I'll very briefly talk to you about some of our very minor contributions which we have made using rice as a system towards understanding the molecular basis or the biochemical basis of abiotic stress tolerance. Especially, I'm going to concentrate on one pathway on which we have been working for many years now, the Glycolyse pathway, which was almost a neglected pathway. Nobody was working on this, but somehow we started working on this, these enzymes. Now, two things become very important if you have to sustain food productivity, besides many other parameters which are important. One is, of course, to increase the yield. And this can be done by various different approaches in order to get better varieties. The second component is where you want to reduce the losses due to the negative impact of stresses. And this could be both biotic stresses as well as abiotic stresses. And your institute has been working on both these aspects and many more for many years. My personal interest has been mainly in the abiotic stresses. And these abiotic stresses, as we understand, will have even more higher negative impact in the future due to change in the climatic conditions. It's expected that we're going to have less of water, energy or land available for agriculture, unpredictable rainfalls, which we already see. Sometimes we don't expect any of these are there. They will get more dry lands or flooding, and now NRI is working on flooding tolerance, on drought tolerance. And also, it is predicted that you will have at least 30 to 35% land area which will become highly saline. It will become unfit for agriculture in future. And this is another interest on salinity tolerance on which we had been working. In addition, higher CO2 as well as temperature variations. It could be very high temperatures or very low temperatures, which we are going to see in specific areas around the globe, as well as in this, our own continent in India itself. Now, there are many ways by which you can tackle these problems. But the last 2 decades, since the genome sequencing of Arabidopsis, rice, and other crops has come up, people are looking forward to using omics approaches, be it the transcriptomics, or proteomics or metabolomics approaches, along with different tools, which could be CRISPR-Cas technologies, transgenics tools, tDNA insertion lines, which people are making. And lately also a great detail of work which is being done on phenomics, and using all these tools and technologies and understanding the basic functional genetic networks which regulate different processes. People want to tinker with the yield components, the developmental control, the quality of seed, and also the stress tolerance, which is our interest. So basically, in order to get better rice varieties, we are looking at using the omics-based knowledge and biochemical tools in order to improve the rice variety. As I moved from JNU to ICGB about 2 decades back, we shifted our attention. I was used to work on light regulation of genes and gene expression and nitrogen metabolism. It was my area at that time of studies. We started looking at to understand molecular mechanisms underlying abiotic stress tolerance. Basically, the questions which were there was in response to stress, how the stress is perceived, and you get the changes in the gene expression, which is both up or down regulated genes. So we've had gene identification through genomic approaches, as well as the candidate cloning of certain candidate gene approach whose functions were known through different methodologies. For getting this differential expression of genes, we also use certain naturally tolerant species, especially Pokali rice, which is tolerant to high salinity. The other question which we have been working, I don't going to get into details in this particular lecture, is the mechanism by which stress activates different genes. What are the signals which are responsible for these upregulation, and the kind of promoters, the stress-regulated promoters, analysis of these promoters, and so on and so forth. And finally, having identified certain genes or pathways, we want to validate these functions, and this is basically done using transgenic approaches. And lately, some of the people in our group have started using CRISPR technology. So linking genes and pathways to stress physiology and tolerance. So these are the kind of broad areas on which we had been working. Of course, now I have retired, my level of activity is much limited, but I will tell you what currently also we are working on. So initially we did some studies with respect to IR64 rice vis-a-vis Pokali rice, and tried to identify the differential genes. So almost like 4,000 genes which were shown to be changing in roots as well as in shoot. This is a comparison between the sensitive IR64 versus Pokali rice. And what we found that these genes which were changing fall into different categories, which are mentioned below the slide, whether these are stress-induced genes, photosynthesis-related genes, signal transduction, so on and so forth. And in many different pathways, these genes were located. We did a little more studies on understanding the regulation of these in a time scale manner. And this is a very, very complicated slide. Complicated in the sense the very filled slide, which normally I don't do it, putting too much of data on this, but this is because of comparisons which had to be made. If you look, what is done in this, on the top of this is IR64. The expression of some of these genes which are listed on the left side here is monitored at different time scales and compared with the Pokali in the different stress conditions. Now, what we see interestingly is that there are many genes which are not at all present in the control, which are the untreated. This is 0 time point, for example. But they get induced as you get the stress response. The only point which I want to make out of this slide is if you look at the Pokali, which is a tolerant one, even at C under controlled, they are already present there. Many of those genes are already present there. Although in some they are also present here, but their expression gets changed. So there are some genes which are induced very early in the response. There are others which are induced later on. But interesting ones are those which are in Pokali, at least, are present right from the beginning. And many of the genes which are listed on the left side, many other members of the group started working on this. But what we shortlisted, although some of these we had been working even before this work started, I will talk about this, but the lessons which we learned out of these, a lot of data was that in response to stress, this is now a very common thing, many other plants also studied, that many genes are up or down regulated. And we have studied in little more details, as I just tried to mention, how exactly they are regulated by looking at the chromatin architecture at the promoter levels of some of these genes, even those which are down-regulated. The second lesson was that induction is kinetically different. Some genes respond very early, the others are very late responders. The induction of genes is tissue or stress specific. And genes that are induced or repressed, they belong to different pathway. It is not that only some specific pathways are involved in this. And last point, which I was just trying to mention from the previous slide, that tolerant species have certain genes which remain upregulated even in the absence of the stress. It looks like that some of those ones, this has been validated in some other plants also, that as if these stress tolerant plants are ready to face the stress, they are already pre-prepared. That's an evolutionary adaptation mechanisms which they have obtained. So out of a large data which we had, many people, as I was mentioning, they started looking at different pathways, different genes, their roles and regulations. My personal involvement was in 4 of these pathways, which is the Gliac pathway, about which I will talk in more detail, this pathway, how we have unraveled this one. The other one, which was Dr. Tuteja's group in our PMB, was Helicase pathway he was looking at, where we overexpress these genes and get tolerance in response to salinity stress. And these genes were later on transferred to rice. But also looking into stress regulated genes, this signal transduction genes, especially the G proteins, G alpha, G beta, and phospholipase C, and a totally novel genes, which are CBS domain containing proteins, on which no work was ever done in plant systems. And it was because of those early profile, we were able to find that these proteins do respond to stresses that, and these are almost like 12 different members of these exist in rice. And we looked into this one, and currently the work by one of the scientists is going on in this direction. Now, what is this Gliac? This is an important slide for you to follow up. Those who have not known about this pathway earlier, this may be an important to follow up the rest of the lecture. Now, what happens that in response to during glycolysis, a byproduct is produced, which is methyl glycol. This is the methyl glycol which is there. Now, this methyl glycol, is a highly toxic compound. Methyl glycol is a very toxic compound. If its level increases in the plant, especially also in rice, and it is also produced in response to lipid metabolism and protein metabolism, it can be a killer. It can kill the cells. It can kill the plants, the tissues at all. Now, this needs to be detoxified. And for the detoxification, you need 2 enzymes. One is Glycolyse 1 and Glycolyse 2. This was earlier what was reported also in the yeast cells and animal systems. They called them GLO1 and GLO2. And this process of detoxification requires glutathione in the process. And after detoxification, the glutathione is returned back to the system, and it can be reused. So there are 3 basic participants in this Glycolyse module. Toxic compound methyl glycol. I will show you later on, it is always not toxic. And 2 enzymes, Glycolyse 1 and Glycolyse 2, and glutathione is used in the pathway. So this is what I call as methyl glycol Glycolyse module in the plant systems. So this whole Glycolyse pathway was actually, if you go to the biochemistry books, not much of it is written in there, but this was discovered somewhere in 1913, Newberg and Dakin and Dudley, they discovered this Glycolyse pathway. lot of work was being done in animal systems. And one of the scientists, St. George, who was a Hungarian but worked in America. When I visited Hungary, I did visit his house where he stayed during early period of his research. He has got the Nobel Prize, and he was the one who really pushed that people that they must look into this pathway. So after that, a lot of activity actually started after he, advocated the cause for this Glycolyse pathway, and its role in many of the disease responses were shown. But no work during this one. A lot of work it was done. Very little work, if at all, was done in the plant system. So this was almost like a neglected pathway. Now, when we started working on this Glycolyse pathway, Glycolyse 1, 2, and methylglycol, somewhere in 1983, our first publication which came. At that time, there was only one publication which was published in Douglas Fir Needles, that Glycolyse activity is there. There was no publication there. And we were looking at from a different perspective the role of these glycolyses. There were a couple of students who did their PhD, Ramaswamy, who is now in USA, Bagga, she also is now in USA, Chakravarti died, unfortunately, worked quite a bit on this. Paulus from Germany came and studied, worked in our laboratory for some time, and she published some work. But it was only around 1999 and 2000 that one of our students, Veena, finally showed that this pathway may have relevance in abiotic stress tolerance in plant systems. It may have a role in stress physiology in plant system, which was not known at that time. Veena also left us, and she has moved to USA. She worked with Monsanto for a number of years, but currently is head of a transgenic research laboratory at Danforth Center. So she has been working for some time, but not in Glycolyse research anymore. Later on, Sneh Parikh joined our laboratory, and she did a lot of work on Glycolyse. And I'm very happy that she's one person who is now one of the leaders in Glycolyse research in plant systems, has been working since then. And after that, many other people came. Yadav joined our laboratory and worked in this pathway. He is currently in CIAB in Mohali as a principal scientist. So many of them contributed. I put this slide to just thank them, all of them who contributed this Glycolyse research. And for a long time, we were the only loners in this pathway. But later on, after 2005, 2006, there were one or two papers earlier, but later on, many other groups have joined and are publishing from Japan, China, and USA, confirming what we were saying. So Veena, when she was working here, what she did was that she had cloned this first enzyme, Glycolyse 1, and she transformed the plants and showed that the transformed plants were tolerant to stress conditions. And that was the paper she published in Plant Journal that time. The cover page came up. And the problem with this single transformation was with one of the member of this pathway, that the plants which were growing, they were not able to set good seeds under stress conditions. There was a lot of seed penalty on these ones. And these initial experiments were done in tobacco. Later on, when Dr. Parikh joined our group, and she cloned also Gly2 gene, and she transformed both Gly1 and Gly2. This was his initial experiments done in tobacco. And when she transformed Gly1 and Gly2, these plants, you see how beautifully they were growing under 200 millimolar sodium chloride conditions. So they were able to tolerate salinity conditions. And not only that, they were able to also set seeds. There were very, very little penalty of 5% or so under 200 millimolar salinity stress conditions. Sneh later on found that these plants, which was published later on in Plant Physiology, is that they're able to also tolerate or grow very well under zinc spike seeds. So basically the information which we are getting from many different sets of experiments, that this pathway may have some important role to play under stress biology in plant systems. The mechanisms by which this happens is something which we worked for, some biochemistry component of it was worked out. And this was just to mention that in 2013-14, they celebrated 100 years of glycolysis research in England. And out of a lot of people I was invited there, but out of many people who gather, there was only one paper on plant. Even after 100 years of research in this particular conference, and Dr. Snee went and presented this paper. So the mechanisms which we felt that time was that under stress, ROS goes up, methylglycol, which is a toxic compound, also goes up, antioxidant enzymes goes down, and because of this, the AGEs are formed, advanced glycation end products, which are very, very toxic to the system, and therefore you get cellular injury and stress-induced damage. Now, what happens in the transgenics, because you have overexpression of glycolyse, which doesn't allow methylglycol to accumulate. So methylglycol levels, the toxic levels remain down. And because of that, because glutathione is involved in the system, GSH levels remain high. We found that antioxidant enzymes are also more, ROS activity is less. And because of this redox balance, which is restored, you get tolerance. So this was a very simplified model which was presented, that in transgenics, they are able to maintain low mg, low ROS, and sustain glutathione homeostasis. Around this time, since the rice genome was totally sequenced, a lot of work was being done in Arabidopsis and rice, we shifted our attention into rice and tried to find out what is the situation of genes in rice. To our surprise, we found that there is not one or two, but there are 11 different Gly1s, the first enzyme in this pathway in rice, and 3 different Gly2 in rice. And similar numbers were present in Arabidopsis. So it's a large family. The Gly1 and Gly2 is a large family which is present in the rice. So rice system probably would be even more complicated. And we wanted to understand what is the function. Are they really Gly, all of these, or do they have the enzyme activities? And if yes, what kind of these activities are there and how they are regulated? So we wanted to clone each of these Gly1s. So slowly, all of these Glys have been cloned over the years, and function of each one of them is being studied. Before that, we also did a study to find out whether you have similar kind of these multiple family genes of glycolase in other systems. And that was a great surprise. And this we have actually done lately. And we find, if you see this slide, just looked at the whole family, this is a bioinformatic analysis in lower E. coli and Heliobacters and bacterial species, and also in animal systems, Homo sapiens, mus, and Ascaris, and Drosophila, you find that they do not have very large families. It is only in the plant where the glycolase family has expanded, both, this computer troubles me a lot. It is expanded in plant systems. Most of the plant systems we have looked at, whether it is mosses, the Fiscometrella, Oryza, Arabidopsis, Brassica, Populus, Medicago, Glycine, you get a large family, large number of Gly1, large number of Gly2, and also there is a Gly3 one, which I will just mention, which is also present in the plants. Now, why do plants have this family, such expanded numbers, and not the other ones? So evolutionary, it should have some significance, and that's what we're trying to look at. So when we looked at these detailed analysis of this in silico analysis, we found that in prokaryotes, you have 2 kinds of glycolases, which is one nickel-dependent and zinc-dependent enzymes. In humans, you have only zinc-dependent enzymes. The glyxlase 1 is only a zinc-dependent enzymes. In plants, you have both nickel as well as zinc-dependent. And nickel-dependent glycolase has 2 domains. Glycolase 1 here also, there's 2 domain proteins which are present here. Glycolase 2 is more or less similar in prokaryotes, humans, and plants. And there is also glycolase 3 in plants we found, which nobody had shown, but this is also in silico analysis. We don't know whether it functions. And it also was something different from prokaryotes and humans. It has 2 different domains, DJ1 domains, which we call it. This also the HS, this heat shock proteins kind of genes which are present. So the question was, what is the nature of this Gliac pathway in rice? Because this is a total plant systems which are doing. So we started looking at the function of each one of these genes one by one. And first question was whether nickel dependent is really present in the plants or not. It took us a long time to identify one Gly1-2 in rice system, which was found to be a nickel dependent enzymes. And it is also inducible by methylglycol. So MG itself induces this pathway. So it is a substrate regulated inducible gene expression which we found. And this has been published, a lot of work in this one has been done to really find out where the nickel binding sites, the protein analysis has been done. I don't want to get into the details of these ones. Another Gly1 gene, protein which we found, which was of course zinc dependent enzymes, but it was localized in the nucleus, again very unique, which was not reported in any other organisms in animal systems or any other one. The Gly2, which we have characterized in more detail, turned out to be a glutathione response to rice glycolase 2, and it's also involved in salinity adaptation and a lot of detailed analysis. This work has already been published. And many other Gly1s, which we are working on, OS Gly1-6, Gly1-10, which doesn't have Gly1 type activity, but it has a unique GST activity. These have been characterized in detail. These manuscripts are in preparation. And earlier we found one glycolase 2, which is a total different gene, ETHE protein, which works for sulfur dioxygenase, and this has been published and calcium regulated. So what we are trying to say that we are trying to characterize the different members of the glycolase family in rice and trying to find out what are their roles and how they are regulated. But more interestingly, as I mentioned in the previous slide, that we found by in silico analysis that there is a Gly3 also present in the rice, which is of this nature. And lo and behold, when we did this work, it was one student who had come from Bangladesh to work in our laboratory, work with Sneh and us, and he was finally able to identify the activity of glycolase 3. Now, what is unique about glycolase 3 is that it does not use glutathione for detoxification of methylglycol to D-lactate. So it's a direct pathway, which is a one-step pathway by which this is done. In fact, this is a very interesting enzyme. And lately, again, I'm not going into details of this, where we have done the whole evolution of these glycolase 3 enzymes, the structural motifs which they have, the functional motifs, and this paper has just been accepted in antioxidant. So a lot of detailed work on this glycolase 3 has been done. Earlier, some group from NCCS and in Bangalore, the US Bangalore, had shown that one of the enzymes of this glycolase 3 can give tolerance when overexpressed in tobacco. Now, as what we wanted now to check, since there is a large number of these families, one of the questions we had, how these different members are regulated under varied environmental conditions. Normally what we do, we'll take one member and we work on it, but we wanted to look at diversity of these members, regulation of these different members at the same time point, how they are regulated. And the second question, of course, don't have much answers to that at the moment, the need for different families in this plant, the glycolase family members in the plant, whether they have any role in development and adaptation. Now, in order to look at regulation of these Gly family members under different environmental conditions, we looked at two important parameters. One is how these are regulated under light and dark conditions. That was my previous interest, to look at light regulation of genes, and whether there is a crosstalk between light regulation and stress. If you put the plants under stress as well as light or dark, and see how these Gly1 and 2 are regulated. And as I mentioned, in the nickel-1 enzyme was also regulated by MG. Does MG regulate these other Gly members? And this is a DBT collaboration work which I have undertaken with Shrudeep, who is a professor at NIT Durgapur. He earlier worked in ICGB, later at NIPGR, but shifted there. But because of his interest in light and my interest in stress, we thought let's combine together and see how we can work it out. And this interest actually followed because we had earlier also published some paper to show that light and stress can, crosstalk with each other. And also programmed cell death under stress conditions is triggered by light. So there were some report from our own laboratory as well as other laboratories which are coming up. And one very interesting paper which might interest some of you is that when the plants are grown under light, the photomorphogenetic state of the plant is dependent upon what kind of a photoreceptor it will be involved. And one of the phenomena which is there, which is called the shade avoidance response, if the plant is growing under the shade of another plant, there is a far-red mediated response. These plants start to elongate so that they avoid the shade. And this is a very important parameter in agriculture. What happens if the plants are facing stress? This shade avoidance response does not occur. It does not grow up, and that is totally blocked. And what was found is that under light conditions, there are two phenomena which are occurring. You either produce brassinosteroid, the hormones, and also the phytochrome pathway, which induce auxins, and auxin then affect elongation. But under stress, what happens that you produce ABA, which block this brassinosteroid pathway, and hence the elongation is stopped. So there is a very interesting, crosstalk between the light and stress responses. Sometimes as a physiologist, we forget these things and we look at only one parameter and not looking at the other. Of course, in environmental conditions, you have many more parameters to look at. So we really looked at these ones. We looked at two genes. One is OS-Gly1-2, which is a nickel-dependent gene. This is a zinc-dependent gene, and this is a Glycolyse-2. And looked at their regulation under light and dark conditions. And this is what we find. And these are the dark. In the light, it goes up. Here, you have initially 2 hours, and then it falls. So the kinetics and the strength of the response varies from the gene on which you are studying. We looked at which photoreceptor would be involved, and we grew these plants under red light or continuous dark or far red or blue light. And again, you find that the responses with respect to each of the member of the families. I'm just giving you data about 2 members of Gly1 and 1 member of Gly2. But we have also done for other members. So I don't want to complicate the slides. So I picked up these 2 because more detailed work was done. And which suggests that the kind of photoreceptor that may be regulating some of the members of these genes families of Gly1 and Gly2 would be also variable. And now when you look at the crosstalk between these members and you give simultaneously when the plants are growing either in the dark or in the light, and simultaneously you are giving them under heat, salinity, or drought conditions, and you find that like under salinity, you find such a great increase here occurring over and above what was happening in the light. And similar responses, depending upon what stress is there, the kind of the stress and kind of the light which you have there makes a difference in terms of the end response which you get. So the whole thing is not very straightforward. It's quite complicated how the environmental signals are perceived and the signal transduction pathways which operate in order to regulate these expressions of the genes. Now, one of the things which we had found earlier, that in response to these stresses, methyl glycol level do increase. This was monitored in different systems, including rice, Penicillium, brassica, and others. In fact, we were the first to really report the endogenous levels of methyl glycol, and to show that methyl glycol levels really increase in response to stress. We had a lot of problem publishing this work. People were not accepting that time. But now many, many more people have done it. And what happens when the MGs goes up? Glutathione level goes down, ROS levels goes up, as I was mentioning, and there is cell death. And there is some indication, as I mentioned earlier, that MG can also induce the expression of these genes, Gly1, and the mechanism by which happens is being studied. So we wanted to check whether MG also differentially regulates different members of the family. And this experiment was done. And you see that depending upon the light or dark conditions, in dark itself, if you give Mg, it shows an increase. These are different concentrations of Mg, and at 2-hour time point, at 6-hour time points, and the responses are different in different members of the gene family. So Mg has a response. It does regulate, and this is kind of a, as I was mentioning, a substrate inducible response, which are very, very few substrate inducible genes present in plant systems. And also, I forgot to mention, there are very few genes which are nickel-dependent enzymes present in plant systems. There's 7, 8 only, and this is Glyvan 2 is one of those nickel-dependent enzymes. Now, since Mg was inducing expression of Gly, we, for curiosity's sake, also looked at whether it works only on Gly, or is it a good signal transduction molecule which regulates the expression of many other genes in rice system. So we compared the expression both in IR64 and Mopokali in response to Mg. So whole Mg responsive transcriptome was studied. In Pokali, there was not much change. It was resistant to changes response to Pokali, whereas in IR64, the number of genes getting changed up or down regulated was very, very large. And we looked at the whole mechanism by which Mg could be acting as a signal transduction molecule. Based on what was reported in the yeast system, we felt that Mg may be working through a MAP kinase pathway in order to regulate the expression of the genes. And we have identified certain Mg responsive elements in the promoter regions of some of the genes, and more work under this is being done. Now, the second question, which is even more important. So what I mentioned so far is that these different members of the Glycolyse family under light and dark conditions, and also under the Mg, are showing a varied response. The question is why you have different members and what is their role in adaptation? I must put my hands up and say I don't have a real answer to these questions at the moment. But the model that I have at the moment, based on the data which we have on nickel and zinc dependent enzymes and their regulation under Mg, stress, and light, I have this proposal. These are all unpublished data at the moment. And the proposal is this, that under normal physiological conditions, where we have light dark conditions, is certain, the width of the arrow shows the amount of the methyl glycol produced. There is a certain amount of methyl glycol produced, and there may be many other signals. So what we find that the level of the zinc-dependent enzyme is always higher than the nickel-dependent enzymes. Its level is much lower. Now what happens? What happens when plant face stress conditions, and also there is light or dark conditions? The amount of Mg produced under stress, as I mentioned you earlier, increases. So there is induction of Gly1-8, the zinc dependent, but not so much. But the amount of nickel dependent enzyme is much higher stimulation, as I showed you under light, as well as under stress. The nickel dependent enzymes shows much higher stimulation. So this is a normally functioning enzyme, but under stress, this gets activated much larger. And if you have much larger amount of the protein which is produced, they are able to then detoxify methyl glycol, and you get stress response or the tolerance response. Now what happens when the stress is much longer? The stress continues for a longer period of time. And for longer period of time, the Mg produced is much higher, which despite the higher production of these two enzymes or even more, it cannot detoxify the system, and therefore the plants will die. So what is the alternative? As I mentioned earlier, the alternative which was shown in tobacco, but I will show it in rice also, that you produce transgenics which are under different promoter, and once you produce these Gly1 and Gly2 overexpression plants under different promoters where the amount of this protein produced will always stay higher, then the plants are able to survive. And this is the model which we have built, and the reason why you have different members and how to modulate these different members in order to get stress tolerance. Now, this is what was shown, that one of the members of this were overexpressed in rice, Gly1 and Gly2. This paper has been now published in Plant, Cell and Environment. You are able to get tolerance to salinity. This was interesting. These plants were able to withstand drought. You can see here under drought conditions, these plants are surviving very well. And when you rewater, then even after rewatering this plant, these wild ones do not recover, but the transgenic ones recover. And they set good seeds. And this is the root phenotype of the wild type and the transgenic plants. We have studied a lot of photosynthetic parameters and that. It also gave high temperature stress and some biotic stress responses. So this pathway, even manipulation of this pathway gave you stress tolerance in plants. We are looking into more molecular mechanisms further of these transgenic plants, looking into their transcript profiling, proteomics, microRNA, this paper published 2 years back, how these are changing and what is really happening inside the system. One of the things which we also found was there was change in trehalose level in some of these plants. This work was being done for a long time, where it's been shown that if you manipulate trehalose by using genes, E. coli genes, trehalose 6-phosphate synthase, TSPP, others, we are able to develop salinity tolerance plants. And this work was done in collaboration with the Karnal Group, Soil Salinity Research Institute, and they have done a lot of analysis of these plants, which were able to tolerate stress. So what message we are getting so far is that one of the things which we published long back, but now based on more work which has been done in rice, we reaffirm that glycolyse and methylglycols could be used as a good biomarkers for plant stress tolerance in plants. But I don't want you to get away with the message that this is a very, very important pathway and this is the only important pathway. I'm sure you also know that one. This is not the, and therefore glycolyse is not the only pathway or the mechanism for salt tolerance. There are many, many other mechanisms by which they are tolerated. Even in our own laboratory, many other genes have been used or expressing in rice, be it phospholipase C, as I was mentioning, NHX and SOS, the transporters. We have actually combined this also with glycolyse and many other genes which have been used. CBS domain protein, which I was talking about, the novel proteins which are there, and they have also been transformed, and the plants showed tolerance to this one. And this glycolyse pathway by many other groups across the world who have taken our clones have transformed into other plants, and they also show tolerance. So glycolyse is not the only pathway. There are many other genes which are also involved in this. And this I've just depicted in this kind of a slide, where many other groups have been using different genes which have been used, different groups, and the end result to being the same. There's a great confusion which comes up. There are many people. There are hundreds of genes. I've depicted only a few of those. Hundreds of genes, individually, different groups, different parts of the world have been transforming rice, and they find that they give you tolerance to salinity or drought stress. So what is the basic fundamental mechanisms behind it? We need to really look at what is that nodal point, if there is any, or they are all working with independent mechanisms. I personally feel there are at least 5-6 important independent metabolic pathways which are involved, especially the transporters, the osmolytes, glutathione homeostasis and others, ROS pathway, which, and at the end, those metabolic pathways become important to give you tolerance. Now, how do you decide, basically with the large amount of genomics, proteomics, and metabolomics, this data which is coming up, which gene is coming? Most of the time we are told that these are complex traits. Abiotic stresses are complex traits. So many multiple genes may be involved in these ones. And this is true, but from animal system, this paper which was published in Cell in 2017, there are many such developmental traits, especially with respect to cancer and others. lot of genes, a lot of different groups are talking about different markers which may be involved there. So what they are talking about that these complex traits, we probably have to move from the concept of what agriculture and what we are talking about, the polygenic traits. And what the concept he's talking about is omnigenic trait. It says the more closely geneticists look at complex traits and diseases, the harder it gets to find active genes that don't influence them. And this is what to me sometimes look like is happening even in abiotic stresses and other complex traits. Many genes people are using and they give you similar kind of end results. So probably it's a omnigenic. Each gene has its certain role to play. Some may have more, some may have less. So whether this omnigenic model will be valuable to use for plant systems needs to be looked at. But what people are trying to do, they are, in order to select these candidate genes from a large, huge, big data, which is coming up on even in abiotic stresses, in developmental traits and other things, they are using artificial intelligence and system biology approaches, using different models, the random forest or other models which are used in order to fish out or recommend which are the best genes which should be used for developing stress tolerance. And based on that, they are then moving into phenotyping screening. So this is AI and machine learning tools are being used in many different laboratories to find out what is really important in order to develop resistant crop plants, both from molecular breeding point of view as well as molecular acid breeding, whatever technology you use. One of the lastly, I question 2-3 questions which I have in mind is, does the whole plant analysis, sometimes when we do this expression analysis, we take the plants which are grown under laboratory conditions, grind the plants, take out the genes and look at the regulations. Does this whole plant analysis under lab, that's the question which I have in the bottom of the slide, or under controlled environmental conditions, do they truly reflect the signature of gene expression? What must be happening under the natural or field conditions? Is it that are we really replicating that phenomena? Because in the environmental, outside environment conditions, there are many other factors which are not regulated. Sometimes the temperature goes up or down, light is many hours of light, many hours of darkness. Nutritional conditions are very different, the soil parameters. So there are multiple factors which are there in addition to the abiotic stresses which we are talking about. So do we really looking at the right kind of genes when we have to develop stress tolerant plants? So the future to me is to understand the plant responses under natural environment. One thing, and secondly, we probably will have to do single cell responses, the single cell omics responses, single cell biology. Because what we do, we take the total roots, we take the total shoots or grind the whole plant, which probably does not reflect because there are a lot of crosstalk even between organ to organ communications are there in stress responses, which we totally neglect at many times. And second thing is we must do field level functional genomics work, which is very, very difficult, but probably an institute like yours, which has all the facilities to do that, should enter into that field. And thirdly, understanding the functions of these genes in ecological context. That's what I was talking, including what is the soil, what is the microbiome, interaction of that, and how does this whole transcriptome really gets modulated. Now, one of these study which was done, especially the field level functional genomics, I will just give one example. This paper came about 2 years back, and that fascinated me. And this was done in sorghum. And this, they have done transcriptome analysis of field droughted sorghum, drought tolerance of sorghum in the field level conditions. Now look at the kind of studies. This was done by UC Berkeley with some other groups. And they have taken 400 samples of the sorghum plants grown during 17 weeks in open fields And from these 400 samples, they have done transcriptomic analysis, and they find at different stages of development which they have done, that 10,000 genes in this sorghum, which is roughly around 40% of the total genome, responses to drought. See the complication we are talking about. I'm just talking about Glycolyse. But in reality, the whole component, that's why he calls it genomic gymnastics. There is a lot more which is happening there. But this kind of work, Limo has mentioned, people have really shied away from this kind of work because they want to do experiment under controlled conditions or greenhouse conditions. But I believe that investment of time and resources is very, very important to understand the real world situation, drought situations. But the amount of effort, the amount of time, and the amount of money required for this was done under like Epicon project. $12.3 million was sanctioned for this project. So whether we will be able to generate that much of resources, manpower, and conditions in which you'll be able to, but this probably to me is the future where we should look at it. Now, once this data came up in sorghum, in the meantime, one of our students, I had been working on sorghum light regulation earlier when I was at JNU. So I thought, let's look at Glycolyse in sorghum. And when she did that work, I think we missed that slide. This paper has been published in BMC Genomics, and she found that there is a large number of, this is the one, where we published this one and found there was at least 15 GLY1 genes and 6 GLY2 genes, and also the third pathway, which is important now, lactate dehydrogenase, are present in sorghum. So this is a detailed analysis, and also many validation was done in this one in sorghum. Now, when this paper came up, I thought, let's look at, I told her, let's look at, this data has been made available only lately, to look at whether Glycolyse is changing under these field conditions under drought in sorghum. And since we had the sorghum paper already out. And when she looked at it, lo and behold, yes, in pre-flowering drought, a lot of Glycolyses, different gene families, they're changing at different time points, both in post-flowering and pre-flowering. And there is a change in the Glycolyses under field conditions also, whether they are involved in conferring drought tolerance in addition to the other things which are happening, probably needs to be validated. But from all this data which I presented in rice, as well as some information generating out of this sorghum, I tend to believe that it is not good to ignore this pathway. And this was one invitation which we got recently, the Tansley Insight Review from new phytologists, where we have tried to show that this pathway over the years since we started beginning, many other groups have taken on it, and its role in different pathway, this developmental issues like pollination under ammonium nutrition and signaling, as I mentioned, it is involved in signaling, in cell death, seed germination we have shown, and many other places where it is being now shown. And we still have to find out what is the function of different members of the family and how they integrate with each other and along with other pathways in order to confer stress tolerance in plant. So with this brief overview of the work which we did earlier and our recent work, I thank all of you for your patient listening. And I also thank all the group members, the former students, the present ones who are working. And special thanks to our DG that time when I was returning from JNU, Mauro Jaka, and Director Dinkar Salunke, who gave me opportunity to come back to ICGB and initiate a small lab, which I very small lab. So I collaborate with Sneh Parikh, with Niti on microRNAs, with Shudeep on light regulation, and with Ashwani Parikh, who has recently moved to NABI as director now. And I thank Government of India for SCRB Distinguished Fellowship Award to me. Thank you very much, all of you, for your patient listening. Thanks.
Thank you, sir. Thank you for nice lecture, sir. With the permission of the chair, the lecture may now be open for the question answer session. The audience may post their queries in the chat box so that they may be conveyed to Professor Sopory.
Okay, there are a few questions perhaps already in the chat box. You can either read out or for Professor Sopory.
I can see in the chat box.
Yes.
Sir,
Otherwise, Suttapa madam
there
can
is one
read out.
question.
Yes.
So there is one question from Dr. Arun Sundaram, sir.
Hmm.
So he wants to know what is the diversity of this glycylase 1 and glycylase 2 in the wild relatives of Oryza?
Yeah, wonderful question. And this is what I have been thinking also for quite some time. I had discussion with N.K. Singh in IRI, who has been looking at the wild rice varieties. In fact, he had done some sequencing also. I actually asked him to give me some data if it's available with him, the genomic data, and we'd like to search for that. We haven't yet been able to get access to that. So as soon as I have access to that data, and any other genomic data available in wild rice, if NRI can help me out on this, I would be the most happy. We really want to look at it because in Pokali we have seen, but not in the other tolerant varieties or the wild rice. So we really would like to look at that. I'm keen on taking up this work.
Sir, so the next question is from Dr. Kaushik. As you have shown both the beneficial and detrimental role of methyl glycylase in plants, how the balance is fine-tuned in rice or other plants?
Yeah, I think what we have some information that the synthesis of glycolase, it is basically because it comes from a glycolysis. And there is an enzyme which is triphosphate isomerase. And that is converting dihydroxyacetone phosphate into Mg. So this is TPI, which changes the balance from DHAP to Mg. And then Mg has to detoxify. So this enzyme TPI and glycolase, their activities will be regulating this the level of Mg in the system. And as soon as you get, in fact, what we have found also that Mg also regulates internally the TPI pathway. So I think basically these are the balance between TPI. We have done a lot of work. One PhD thesis was done only on TPI in rice. And so this balance of TPI and glycolases, which will regulate Mg. Now, Mg becomes just like ROS. They say at lower concentrations, below a certain point, it becomes a signal molecule. So this was a paper which was published in Nature long back about ROS, about 3-4 years back. Very interestingly, they showed the balance of ROS at which it becomes signal and balance of Mg, which will act as a signal. So this has to be maintained by these two enzymic systems. And in stress, this gets disturbed. If a long-term stress, this gets disturbed and you accumulate too much of Mg, which becomes a toxic. And therefore, that's why we thought the transgenic keeps this balance back to the minimum level. These things still need to be understood in more detail.
Thank you, sir. So there is another question from Dr. Parimalan Rangan. He wants to know that what is the role of transporters in this?
Well, transporter plays a very important role because sodium homeostasis has to be maintained by transporters, both by sauce-like membrane-based transporters, as well as NHX transporters, which are on vacuolar membrane. We did some work on sauce and NHX, and we feel that that is another pathway which has to play very important in maintaining homeostasis. Now, glutathione also, because Mg pathway also maintains the glutathione homeostasis. And we had some indication, many other groups also shown that glutathione itself can act as a signal molecule. Now, whether glutathione itself regulates other homeostatic pathways, ionic pathways, needs to be looked at. But in sodium homeostatic pathway, I think potassium plays as much as role. If the HKT transporters are also playing an important role. So it's basically sodium potassium homeostasis which needs to be maintained in order for plants to tolerate these conditions. Ions become very, very important in this whole sodium salinity tolerance, at least. I cannot say for the drought may not be as much, but for salinity, yes, that is an important parameter.
Thank you, Professor Sopory, for patiently, very elaborately satisfying different queries raised by the audience. Thank you, sir. Now, I would like to request our director and president for. Yes, sir.
Director, sir, kindly give the president's remark.
Okay. If there are no more questions, okay. Thank you, Dr. Shutapa, Dr. Patra. It's really a very good afternoon to one and all. Respected Professor Sudhir Sopory, Honorable former Vice Chancellor, Jawaharlal Nehru University, the August gathering on this virtual platform, senior officials from ICAR headquarters, my colleagues of National Rice Research Institute and other institutions, my dear students, ladies and gentlemen. It's really my great honor and proud privilege to listen to Professor Sopory, the world-renowned plant physiologist, and also our honorable chairman of Research Advisory Committee of this institution, and his deliberation to topic Glycolyse pathway in rice, its role in abiotic stress tolerance. As you know, Professor Sopory's areas of research specialization are molecular plant physiology, stress biology, and plant biotechnology. As we know, the several abiotic stresses are very important constants in rice, as this crop is grown in almost all types of ecology and thus faced several abiotic stresses, and sometimes more than one stress are being faced by this crop in single season at different growth phases. So imparting identified multiple stress tolerance in rice varieties have become very important to stabilize yield across ecology. Thus, scientists look for all possible ways to address this complex issue to make it more robust. And one of the ways is overexpression of glycolyse enzymes involved in glycolyse pathway, as Professor Sopory educated us on this aspect. And it detoxifies the methyloctase, a cytotoxin produced during abiotic stress, particularly in case of salinity stress, and thus imparts tolerance. So Professor Sopory's deliberation created an environment of learning, radiating knowledge to all corners, and ignited the scientist's mind with several innovative ideas, which is apparent from the lively interaction session we have just witnessed. So I extend our gratitude and thanks to Professor Sopory, sir. And sir, I expect our scientists to have the opportunity to interact further physically during our next RSC meeting. So thank you very much, sir. Thank you all.
Thank you. Thank you very much. Thanks for giving an opportunity. I wish one could interact personally, but these are the limitations of the time.
Yes, sir.
To work like that. Thank you, everyone.
Thank you, sir.
Thank you very much, sir.
Thank you for your valuable remarks.
Thanks, everybody. All the best.
So now we come to the end of the today's lecture. Hence, it's time to express our gratitude to all who have contributed to make this event a big success. For the purpose, I, on behalf of my institute, take the opportunity to wholeheartedly thank our today's speaker, Professor Sopory, for accepting our invitation in spite of your very busy schedule, sir, and delivering the lecture on the Glycolyse pathway in rice. Your talk has been very educating You started with very basics of the Helicase pathway, moved on deciphering the discoveries in a storytelling manner that could carry the audience of diverse faculty along the journey with you. Sir, you talked about the pathway, the genes involved, the cross-talking among genes, the gene families, and many other complex things in a very lucid manner, which can be understood by all. For this, I express hearty congratulations to you. I'm sure the lecture could ignite the faculty in many of us working in similar areas. Then I would like to express my sincere thanks to our director, Dr. Maiti, for his continuous support and encouragement for conducting month-long series of lectures to mark the Platinum Jubilee Foundation Day. Thereafter, I would like to thank all the heads of divisions, OICs of regional stations for their support to organize the event. Then I would like to thank Chairman of the committee, Dr. B. Satra, for successfully coordinating the activities towards organizing lecture in the series. Thank you all the members of the committee, Dr. Kutub, Dr. Mohanty. Without your support and cooperation, it would have not been able to organize the program. I express my sincere thanks to all the OICs, sincere thanks to OIC Erisel for wide circulation of the event, and special thanks to Mr. Santosh Kumar for generating the Zoom link and providing logistics to conduct the event without interruption. Special thanks to all audience from National Research Institute and different other institutes, students and researchers who got connected in large number for the event and actually participated in the question answer session. The total number of participants this time also touched about 100 to make it a success. Thank you to one and all who have directly or indirectly contributed for the success of the event. Thank you and all for the patient listening. With this, we come to the end of today's event. Thank you and all. Have a nice day.
Thank you very much, sir.
Thank you. Thank you.
Thank you.
Yeah, thank you. Namaste. Sir, you can close now.